we have completed 16 sessions, which we will call part one. And today we will be entering part two, sort of part two of the exploration of awakening awareness, of investigating our being. And we've come a long way. We started all the way back with resourcing, which is in my opinion, one of the most important things to really have down. And so it's really important from my perspective in this practice that as you move along, that you feel comfortable with resourcing, that you understand what it is, that you have a practice that is based in resource. And so if anyone came and asked you, do you have a resource, that it would just spontaneously sort of appear, or that you practice it so frequently that your resource now is not something that you need to think about or bring on, but it's just there. It's like, oh, where are you resting? Well, I'm 95% of the time I'm resting in my resource. Well, what is that? Right? It's a place that I can direct my attention to, that I feel comfortable, or it's a sensation, or that I feel safer with, or it's an image, right? And that slowly, if it's not in our body, we actually use the process of titration, pendulation, and resourcing. And that resource might be something outside of our body, but that slowly we bring in our body with being able to feel the felt sense of our body. And that can be through a slow titration process. And I've spoken about this in some of the previous lectures. And then really becoming familiar with the breath. Going from, if the breath is not your initial resource, that you might use your resource to engage the breath. The breath might not be a comfortable place, a place that you feel safe initially. And so therefore you can do a titration or pendulation from your resource to the breath, and slowly bring on the breath, because the breath is a very, very powerful trigger for the parasympathetic nervous system to help balance the fight or flight response, the autonomic nervous system, what we call it, and to help increase vagal tone, because the vagus nerve is the nerve that it's the main driver of the parasympathetic nervous system, autonomic nervous system. And so we need exercises to actually activate that, because we are living in a world that is constantly activating our sympathetic fight or flight survival. And so we practice with the breath, and it's an automatic switch for our parasympathetic nervous system. And then we came through having an understanding of awareness, looking at our senses, sight, smell, hearing, touch, taste, and then going inside and noticing our senses on the inside out, the more subtle senses of what does our heart feel like, what does our lungs feel like, our gut, what are the various, if I was to perceive the world from the inside out, what would it be like, as opposed to constantly perceiving it from the outside in. And then going into the activities of the mind, including thoughts, emotions, beliefs, and really seeing the separation, noticing the thoughts and emotions as emanations, creations from awareness, from this plane of potentiality. And that in essence, although they seem distinct and separate, they're really the same thing. And so then sort of having a practice of being able to rest in awareness, fully embodied, feeling yourself seated, and noticing what that is like for you. and that our common plane, our common playing field is awareness. But there's the awareness that is unifying all of us, that the awareness that perceives your experience in this moment is the same awareness that is experiencing my experience at this moment. And so there's this interconnectedness, and with intention, we can actually go into that interconnectedness and just sense that interconnectedness of all beings. And that then can become your resource. Okay. And that you practice this enough so that it's like you're learning a new language, and next thing you know, you've practiced it enough, you've repeated enough, you've done it enough where you're not only training your mind, but you're also training your heart, you're training your body to have this felt sense. And so you can sort of come home. It's easier to come home more frequently. And your resting point, your zone of, let's say, neutrality, is pure awareness. And that's then where all things emerge from, including what you choose to say or what is spoken, what is experienced, what is felt, right, what is heard, et cetera, et cetera. And so now we enter part two, which to me is an integral part of this dynamic relationship of really the unmanifest and the manifest, the unmanifest and the manifest, undifferentiated consciousness to differentiated consciousness, the plane of all possibility to that one thought, let's say that one creation. Hey, I want to make a new airplane. Well, that's how it begins, right? It's a spark. It's a wave that emerges, and this is how everything is created. Everything is created, and so there's this relationship then between the unmanifest, pure source, pure potentiality, the plane of all possibility, and manifest. And that manifest can be a thought, it could be a spoken word. It's anything, any object that we experience is a manifestation. When we are in meditations, one of the things that helped me investigate this movement of energy, if you want to call it Kundalini, if you want to call it awakening, whatever you want to call it, was actually to have a fundamental understanding of embryology, where we came from as physical beings. Okay, so when I entered this, I actually it was just by chance that I studied. My major at UCLA was molecular, cell, and developmental biology. And it had just been created as a major because I was very fascinated in how a single cell becomes a multicellular dynamic organism. And so you can imagine the number of classes we had in terms of what we call developmental biology, the embryological formation of physical form. Okay, and you'll see kind of how this plays, hopefully over the next few weeks as we dissect the embryology a little bit. For me, the importance of this is that when you are in a meditation, when you do feel as if you are resting in awareness, what you may notice, what you may have the experience of, now again, this is an object of that is emanating, but you may have this experience of embryological movements. Now, it's not to get excited about them or to become attached to them, But when there is a knowledge, for instance, about the possible embryologic movements, in my experience, it's not something that like uh, you get lost in. You can simply notice it and start having a relationship with it, in essence. So just like any object that arises, you can witness it. You can have this relationship, for instance, from, let's say, the ocean going up into the wave, creating the wave, dancing like the wave dances, understanding the wave, and then the wave comes back into the ocean. It's a similar process of resting in awareness. And as a movement, you may actually feel a movement in your body now that was an embryological movement that you did many years ago in the womb. You go, wow, this movement just sort of seems to be, and I don't know, I can't, but we're not like, it's not like an over analysis. It's just sort of a noticing. It's sort of like, oh, this movement is coming up again. I'm here and I notice this movement. And you just sort of sit with it and ask, what could it be? Similar to the question we had earlier of, could there be a vibration that is actually causing sound that is coming through my cribriform plate? Could it be? And you sit with that and you notice it, right? But we don't get too attached to it where that's the only thing that we're paying attention to. We're still sort of resting in awareness. And so what there is this ability to be aware of awareness itself while we're also aware of what is emanating from awareness simultaneously. So we're sort of sitting, resting in awareness while we're also aware of any interesting movements that may start appearing or felt senses that may start emerging in the body that we can actually then consciously, and this is sort of the important part, is through awareness consciously placing an intention into that movement and saying, oh, what is this? What is this? As we place an intention into the movement, we actually start to understand the movement itself. And it feeds back. It's sort of like this movement then, as we consciously bring awareness to something maybe that occurred embryologically, we're not only allowing it to maybe complete its movement or fulfill its movement or to feel it happen, but there's this concept of, as we bring awareness and consciousness to something that we are actually feeling in the body, and it's let's say an evolutionary movement of energy that it can add to, it can activate slightly that movement of energy to allow it to flow and to continue flowing just like energy should flow, as opposed to feeling as if it's blocked. When we allow it to then flow and move through using a bit of conscious intention, that then is informing our mind, and it sort of creates a positive feed forward loop, sort of like this positive feed forward loop. Consciousness, or our mind, will become aware of something that is occurring in our body with pure awareness and neutrality, setting a little bit of an intention for it to actually move and move through us, but from the plane of awareness, which isn't getting caught up in the movement, it's simply allowing it to flow. You're not getting scared. You're not going, oh, but you're also not getting excited. You're not like, oh, you're like, oh my God. You just allow it. There's just this allowance, but it's a conscious intention, right? So like, oh, let me notice. I'm really noticing that, and I'm totally with it, right? And as you bring conscious awareness to it, you can actually allow it. It's almost like you're supplying it energy from this place of awareness that you're choosing to, allowing it to move, and we're not getting caught up in the emotions or anything. If emotions arise, they simply arise. You might start crying. Great. Don't get too caught up in it. Right? And so there's this sort of feed forward loop as we bring conscious awareness to something that may be moving in our body that might be subtle as it might be, that the subtle energy recognizes the conscious awareness, that then the subtle energy has a greater propensity to move and flow, which then feeds forward to the brain, the mind, and it continues this loop. And this is a loop of body and mind evolution. And so when we, being embodied, in my opinion, has a prerequisite of also understanding your anatomy to some degree, and to understanding your embryology, how this body actually formed. Where was the source? And we could go back, and we will, go back and look at DNA. But at first, to get a sense of, even if we ask the question, for instance, well, where's this thought coming from, right? Where's this thought emanating from? Well, look, why don't we go back and take a look at like where this arm came from, or where this leg came from, or where my heart came from, or where my, my head came from. Like, where did it, where did that come from? And so I hope that makes sense to some degree. I have a video that I want to show, and I want to go through this video. And these next two lectures, well, this one for sure will probably not have a meditation associated with it. But these next two lectures are really going to look at embryology. And I have a video that I want to go through together. I'm going to share my screen. And if you want, this is probably the best, shortest video I have found in terms of putting it all together. And so we're going to go through it and kind of dissect it together. All right. So take this in, right? And just like anything, this is how we were all created. Okay. So just imagine, as you're listening to this, that this is how you were created. All right. And as I feel like there's necessary things to sort of point out, I will. All right. So it's got to start off with fertilization. It's got to start off. All right.
When ejaculation occurs during intercourse, approximately 200 million sperm, or spermatozoa, are deposited into the vagina. They swim through the cervix, propelled by whip-like motions of their tails, or flagella, after which muscular contractions of the uterus direct them to the uterine tubes.
Okay. So first of all, 200 million. Okay, that's a lot, right? That flagella, the flagella-like movement, you may actually have a sense of that. You may actually have a sense of a flagella-like movement in some of your meditations. You may also have a sense of the uterine contractions that are performed by the uterus totally spontaneously when it has sperm inside of it. Okay.
This process usually takes between 30 minutes and 2 hours. Only around 200 spermatozoa will reach the secondary oocyte in the...
So 200 million to 200. That's quite a decrease in number. All right.
uterine tube, and of these, only one will fertilize it. Fertilization cannot occur until two processes have taken place: capacitation and the acrosomal reaction. These can take several hours. Capacitation is not fully understood, but secretions from the uterus wall and uterine tube destabilize the plasma membrane surrounding the head of the spermatozoa.
All right, so out of the 200 sperm that get up there, so there has to be secretions from the uterus that destabilize the sperm. So already you're talking about a relationship that is occurring between the fluid that is present in the uterus and the sperm. Right? So there's got to be a relationship. There's got to be some fluid that's saying, "Hey, I am going to destabilize you such that you have a greater chance of getting in." Okay.
Or acrosome, resulting in the membrane becoming more fluid, which helps to prepare the spermatozoa for the events of fertilization. The spermatozoa become hyperactive. Their flagella beat more frequently and their heads move laterally. The capacitated spermatozoa move through the corona radiata, a dense layer of granulosous cells surrounding the oocyte, and come into contact with the zona pellucida. The zona pellucida expresses specific receptor proteins called ZP3, which bind to proteins expressed in the heads of the spermatozoa. The binding of ZP3 triggers the acrosome reaction, during which the enzymatic contents of the acrosome are released. These enzymes help to digest a path through the zona pellucida, allowing the spermatozoa to enter the perivitelline space and reach the plasma membrane of the secondary oocyte with which it fuses. To ensure that only one spermatozoon penetrates the zona pellucida and fuses with the oocyte membrane, fusion of the spermatozoon and oocyte membranes activates a fast and a slow block to polyspermy. During fast block to polyspermy, after fusion, the oocyte membrane depolarizes, preventing other spermatozoa from fusing with it. Slow block to polyspermy is also stimulated by this depolarization. During slow block to polyspermy, a wave of intracellular calcium is released, causing small cortical granules beneath the oocyte membrane to release their contents, rendering ZP3 inactive and making the zona pellucida impermeable. Upon the spermatozoon entering, the oocyte undergoes meiosis II and further develops into the female pronucleus. During this time, the sperm develops into the male pronucleus, and the two pronuclei fuse to form a single diploid nucleus, or zygote.
All right, so then they have a really good summary in terms of what they just talked about, the process of the sperm coming into the egg, right? The process then of once a sperm fuses with the cell membrane, that it triggers these two reactions that help prevent other sperms from coming in, right? So just think of the dynamic relationship that needs to occur not only from the membranes fusing and becoming one, so that then the sperm can put the DNA, what's in the nucleus, into the egg, but also the calcium release. Okay, these are all energetic processes that are occurring in order to allow one sperm in, but then to stop the other sperm from coming, so you don't have 10 sperm or 20 sperm, let's say, that enter the egg at the same time.
After fertilization, the zygote undergoes rapid mitotic division, known as cleavage. The first cleavage, completed about 30 hours after fertilization, produces two identical cells called blastomeres.
So what happens is that the DNA from the egg and the DNA from the sperm, right, come together, and 30 hours after fertilization, they come together, right? And that's when you get mixing of your DNAs, and one cell, the cell which was the egg, has been fertilized by the sperm, now can actually cut itself in half, essentially. It creates double the DNA, and we'll go into that in the future. It creates double the DNA so that one whole part of the DNA can go to one cell and one whole part of the DNA can go to the other cell.
This cell division continues, and by day 3, there is a cluster of 16 identical blastomeres. This is known as the morula.
So 3 days, right, to get this one cell that has sperm and egg DNA in it, 3 days to get to the 16 cell stage. 16 cells. So what divides once, now you have 2 cells, divides again, now you have 4 cells, divides again, you have 8 cells, divides again, you have 16 cells. Right? At this stage, you can take out one of these cells. If you go in a lab, you can take out one of these cells from this 16 cell stage and create an entire new human being. Okay. So these cells are called what we call totipotent. They have total potential, totipotent. Okay. When you're not totipotent, you have words like pluripotent, where you have many potentials, but not all potentials. Right. So just think of it from an awareness perspective, right? Awareness, everything is totipotent. Everything is possible. Okay. So at this cell stage, still, after 3 days, every single one of these cells, if I took it out and put it in a petri dish, I could make an entire new human being. Right?
The morula enters the cavity of the uterus around day 4. At this point, around the 32 cell stage, a fluid known as uterine milk starts to penetrate the zona pellucida to nourish the blastomeres.
Whoa. So there's no blood supply, right? The egg is deposited into the fallopian tube. There's no blood supply. The egg is separate. Okay. As it gets fertilized, there's still no blood supply. The egg itself has all the nutrients that it needs up until day 4, when it absorbs this uterine milk to give it some nutrients and to have fluid now enter the cavity. Okay, so the egg, this now fertilized embryo, needs to be receptive to taking in that fluid to continue being viable. All right. So this was you. You had to be receptive to taking in this fluid for you to continue this viability, because you were essentially isolated from anything. You are not in the uterus. You are no longer in the ovary, and you're cruising down the fallopian tube. And essentially, you could just go in there and take this out, and that would be the end of that fertilized embryo. Okay. So that fluid comes in, it provides some nourishment to it, but you need to be willing to take it in. Oh, there it is. And notice I'm absorbing this fluid. Okay. Just think of that receptivity, total acceptance. Okay. Here I am. Wow, what is this? What is this coming from the outside? Looks like a storm. Oh, it's uterine milk. Let's just let it in. When's the last time you were bathed in uterine milk? Well, it was 46 years ago. You know.
Blastomeres continue to divide, and as more uterine milk enters the morula, they develop a central fluid-filled cavity, known as the blastocyst cavity or blastocele. Around day 5, after the development of the blastocyst cavity, the developing embryo is known as a blastocyst. Around this time, the zona pellucida degenerates and the blastocyst hatches into the uterine cavity, ready to implant into the uterine wall. Implantation occurs approximately 6 to 7 days after fertilization, but can only occur if the endometrial wall is sufficiently prepared by the correct levels of hormones. The blastocyst usually implants in the posterior portion of the fundus of the uterus. Initially, the blastocyst attaches loosely to the endometrial wall, but this attachment becomes stronger as the blastocyst burrows into the endometrium, and the endometrium becomes increasingly vascularized. Eventually, the blastocyst is completely embedded within the endometrium, at which point the endometrium becomes known as the decidua. The regions of the decidua have specific names relative to the site of blastocyst implantation. The area between the embryo and the stratum basalis is known as the decidua basalis. The area between the embryo and the uterine cavity is the decidua capsularis, and the remaining area is known as the decidua parietalis.
Okay, so if you're doing this, the only error I found is cleavage begins 30 hours, not days, after fertilization. So that's to be hours. The other important part is that the blastocyst implants into the wall after 6 to 7 days. 6 to 7 days. All right, so that's how long. The let's call it the fertilized egg now. That's how long it is outside, essentially its own entity, going down the fallopian tube and trying to find an area on the uterus that it can implant in. All right. So there has to be this ability to bring in the uterine milk to supply it some nutrients. That is fluid found within the uterine cavity. All right. There also needs to be, as it said, right, and this is sort of a beautiful relational perspective that may come in your meditations as well, is this the uterus has to be ready. It's got to be prepared. It's got to be a welcoming environment for the this fertilized egg to implant. If it's not a welcoming environment, if it's not ready, if it's not prepped, that fertilized egg gets peed out. That's it. Okay. So just imagine every single one of us was at this point where we hit the uterus wall, and our mother, the uterine wall from our mother, decided, was ready for this relationship. Maybe it had never seen this egg, had never seen this before, right? Imagine you're in, I don't know what that emotion, I never experienced that emotion. Here's the uterine wall seeing this for the very first time, or maybe it was the second time or the third time, seeing for the very first time, and the uterine wall being so prepared and nurtured and ready that as soon as it noticed this new being that comes in, it's totally primed to say, "Come, I'm here. I hold you." Right? This happened in all of us, in all of us.
Implantation of the blastocyst usually occurs 6 to 8 days after fertilization. By the end of day 8, the blastocyst has burrowed into the endometrium of the uterus. At this time, it is composed of two main components: the outer cell mass, the trophoblast, and the inner cell mass, the embryoblast.
Okay, so this is really key. Where we develop from, the cells that we specifically develop from, are from this inner part, the embryoblast. So you see how it's essentially a single cell layered film. This is where we all came from. As we are embedding, as there's this mutually beautiful relationship between the fertilized egg that's coming into the uterus accepting it, bringing in this egg, that which is going to develop into you are those blue cells that you see, one cell layered thick. Imagine that. Imagine that.
As the trophoblast makes contact with the endometrium, it differentiates into two layers: an inner cytotrophoblast and an outer syncytiotrophoblast. The embryoblast differentiates into a bilaminar embryonic disc composed of two cell layers, the hypoblast and the epiblast.
Right? So it goes from this single cell layer to two cell layers. Okay, do you see that? As it's entering, what is the egg? What is this embryo starting to feel? I have nourishment. There is receptivity. There is acceptance by this fluid, by this tissue. I can continue to go through this process. Right?
Soon after the embryonic disc has formed, a cavity begins to appear between the epiblast and the cytotrophoblast, known as the amniotic cavity.
Okay, remember this image. All right, you see where the amniotic cavity is? That's the amniotic cavity. Tiny. But that's you developing right next to it, the blue and the yellow. Okay? Just keep that image sort of, watch this video. This is at 7:26.
Cells originating from the hypoblast begin to migrate, forming a thin membrane, which covers the inner surface of the cytotrophoblast. This is called the exocelomic membrane. The exocelomic membrane and cells of the hypoblast together form the walls of the primitive yolk sac. By day 9, the blastocyst is completely embedded in the uterus wall. At this stage of development, the growth of the syncytiotrophoblast and cytotrophoblast is much quicker than the bilaminar embryonic disc. Small holes called lacunae begin to form in the syncytiotrophoblast as it continues to expand. By day 12, the lacunae stop growing and fuse to form large interconnecting spaces called lacunar networks. Capillaries in the endometrium surrounding the developing embryo dilate, forming maternal sinusoids. As the syncytial trophoblast continues to expand, enzymes begin to erode the lining of the sinusoids and uterine glands, allowing maternal blood and uterine secretions to flow into the lacunar networks, establishing a utero-placental circulation. The blood and uterine secretions only come into close proximity to the embryo, allowing the exchange of gases and metabolites. Around the same time, a new population of cells appear between the inner surface of the cytotrophoblast and the outer surface of the primitive yolk sac, known as the extraembryonic mesoderm. Large cavities begin to appear in the extraembryonic mesoderm. These gradually fuse to form one single cavity, called the chorionic cavity. Around 13 days after fertilization, a large portion of the exocoelomic cavity is pinched off, forming a smaller cavity, the secondary yolk sac. By the end of the second week of development, the chorionic cavity enlarges, and the bilaminar embryonic disc is joined to the trophoblast by a band of extraembryonic mesoderm called the connecting stalk, the future umbilical cord.
All right. So here you are. You're still a bilaminar sheet, 2-cell layered sheet of cells at day 13 or day 14. You have a connecting stalk that is going to become your umbilical cord, which is your umbilicus. So when you touch your umbilicus, energetically, you are touching this stalk when you are an embryo. All right.
Called the connecting stalk, the future umbilical cord.
Okay. So we're going to finish the rest of the video next week. But that was, so some of the language, if you rewatch the video, the words, those are just, different words for different cell types or different cell layers that are forming. I would probably review it maybe once or twice and go over it yourself, because this is really where we all started out. And if you think about it, back at day 14, all these relationships, all this stuff that had to happen for us to be manifest into physical form. Right? If at any one point the uterine milk wasn't enough to supply the nutrients, out we go. If the uterus didn't accept the fertilized egg as it's coming down, if it wasn't ready, primed, if the tissue wasn't, out we go. If the egg is going in, fertilized egg is going in, and there's not enough lacunae that form, right? Those spaces, and that's the only thing that is bringing nutrients to the fertilized embryo. Okay? And as they say, the blood capillaries fuse with this lacunae, and imagine like you're going from like lake to lake or cavity to cavity, and there's like blood that's sitting here, and just by the diffusion of gas and metabolites can the fertilized embryo actually get any nutrients. Right? But all that already needed to occur as we're sitting there, and as the cells around us that are creating these cavities are expanding. And so you may feel that expansion as we are slowly creating this bilaminar disc, okay, even at day 14. At day 14, you're still two cells on top of each other, two cell layers on top. Go make like a cake. Make it make it two layers thick, and then take the cake out and go, all right, there's a vanilla layer and there's a chocolate layer. And you look at it and go, wow. Now these layers, look at the cake. These layers are going to form me. You go, yeah, yes, yes, they are. And then you sit back and you go, oh my goodness, this is amazing. This is amazing. And I get to taste pizza. Yes, you get to taste pizza and you get to smell flowers and you get to see the color purple or red or green. It doesn't matter, but you get to hear music. You get to create your own music. Maybe it's out of your nose. Who knows? You feel, I feel like when I look at this, it's sort of like the energy just buzzes. It's like, this is incredible. And just simply resting in that is like awe, right? And you're just like, oh my goodness. And I'm getting caught up with like my thought of like planning an appointment on Friday. Oh my God, do I make the appointment at 1:30 or 3:30? But then I have to take the kid, it's like, whoa, it's like, hey, you know, why don't can we allow that same energy that allowed the sperm into the egg, that allowed that allowed the cells to divide, that allowed the relationship to occur between the fertilized embryo and the uterus. Can we just allow that same energy at work, even today? Can you allow the same energy that took you as two cell sheets to manifest what is to manifest for you? Just sort of. Whew.